Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is obviously endocytosis. And here in my previous video I have explained about the Caveolae Mediated Endocytosis, and today here I will try to explain about the Caveolae Mediated Endocytosis. Okay. So before going to the topic, I want to say that please you subscribe and hit the bell icon of my channel for getting the notifications of all my uploaded videos. Okay, so let's start. This is the Caveolae Mediated Endocytosis. What is the Caveolae Mediated Endocytosis? First of all, we have to know about the caveolae. What are the caveolae? These are the flask-shaped invaginations on the cell surface in many vertebrate cell types. So, this is very rough structure. You can see that it is the flask-shaped structure, okay, of plasma membrane, or it is seen just on the cell surface. This is called the caveolae, okay, and these are localized, or these are found in generally endothelial cells and adipocytes, okay. And these flask-shaped structures, there are many proteins and also lipids, okay, and under lipids main cholesterol and sphingolipids are present, and proteins means transmembrane proteins, they are also present. The formation and maintenance of the caveolae is primarily due to the protein caveolin. Okay, so caveolin, it is one kind of molecule that actually maintains the maintenance of the caveolae, or the formation of the caveolae, that is maintained by the caveolin. So what I am saying that caveolin is one kind of protein that helps to form or maintenance of the caveolae. Clear? So these are the proteins. Caveolin, these are the proteins, and that helps in the formation and the maintenance of the caveolae. And this protein is an integral membrane protein. Okay. So what are integral membrane proteins? This is actually these were explained in my previous video about the structure of the plasma membrane. So you can check out for these queries. Okay, what are integral proteins? Now, what I am saying that caveolin, these are the integral membrane proteins, and they have C and N, both terminal sites, okay, and these are actually present where? In the cytoplasmic site, okay. Suppose this is cell and this is the plasma membrane, this is the plasma membrane, so this is the cytoplasmic area, okay, these are the totally cytoplasmic area, this is the cytoplasmic area. So this C and N terminal sites, or C and N terminals, that remain like that inwards to the cytoplasmic way, okay, not to the outward side of the plasma membrane, just towards the cytoplasmic area. Next, what is that? 3 palmital groups attached to the carboxyl terminal globular domain further anchor it to the membrane. Okay, not only C and N terminal site present, or C and N terminus you can say, also present 3 palmital group, okay. This palmital group that are attached to the carboxyl terminal site, or carboxyl terminal domain, globular domain, okay. So what are the terminal sites? One is C, another is N, and third one, that is the this is the terminus, C and N main terminal site, and 3 palmital groups, these are present where? The carboxyl terminal region, okay, carboxyl terminal region, and this is generally remain, these are remain for further anchoring with the plasma membrane, Okay, so main structure of caveolae is that these are actually the integral membrane protein, and they have C and N terminus site and that remain at the cytoplasmic site. Also 3 palmital group attached to the carboxyl terminal globular domain. Okay. Caveolae, next we will learn about the process, how the endocytosis is occurring. So caveolin that binds the cholesterol in the membrane. Okay, there are cholesterol present in the membrane. So caveolin first bind with the cholesterol, and the presence of caveolin that forces the associated lipid bilayer to curve inward and forming caveolae in the surface of the cell. Okay, so how these caveolae are formed just surface of the cell, how it is possible? First, caveolin that binds with the cholesterol lipid, okay, and this in presence of this caveolin, this association of the cholesterol and caveolin forces the bilayer to curve inward. Okay, that means like this and this portion. So this becomes curve and to form the caveolae. This becomes curve. This membrane surface region that curves to form this caveolae. Okay. So how is this possible? You totally understand what I am saying, that caveolin first bind with the cholesterol and then forces to form just curve-like structure inward the cytoplasmic area. Inward the cytoplasmic area. Okay. Now caveolae is formed. Okay. And now caveolae pinch off from the PM. PM means plasma membrane. This caveolae first formed, then pinch off from the plasma membrane and just form a spherical structural vesicle, okay, or spherical structural molecule you can say. And this pinch off from the PM, how it is possible? With the help of dynamine. Dynamine, one kind of molecule, and this is also one kind of protein you can say. So it helps in the pinching off of the caveolae from the plasma membrane. Okay, so caveolae containing material, this material, what happened to this material? These materials generally deliver their contents either to an endosome like compartment, this is called the caveosome, or to the plasma membrane on the opposite side of the polarized cell. So either it form caveosome like structure or it form one kind of compartment that is called the caveosome, or what happened that PM on the or release this contents on the opposite side of the polarized cells. Okay, I am saying again. What is that? They deliver their contents either to an endosome, that is nothing but the caveosome, or on the opposite side of the polarized cell. Okay. Now caveolae contains some receptor proteins and are used for certain types of receptor mediated endocytosis. Okay, this is another thing, that is sometimes caveolae they have receptor proteins and there the endocytosis is possible with the help of the receptor proteins, and generally receptor protein containing endocytosis is actually explained in the Caveolae mediated endocytosis. Okay, so this is the rough structure of the Caveolae mediated endocytosis. Okay, so this is one kind of Caveolae mediated endocytosis. Okay, and this is the topic, how the endocytosis is possible with the help of the caveolae. Okay, and now we will learn when the caveolae is absent or clathrin is absent, then endocytosis, how is endocytosis possible? Okay, so let's continue the next topic. Okay, so clathrin and caveolae independent endocytosis, that is actually specialized high capacity endocytic pathway, and this is a pathway for what? Lipids and fluids. Okay, so this is found in the primitive stage. Okay, so if this type of endocytosis is found, as it is found in the primitive stage, and this is occurring just for lipids and fluids, so obviously with a specialized high capacity way, so this is totally another kind of endocytosis, and as it is found in the primitive stage, so here it is not explained properly. Okay, so in this book, actually from where I get the information, so there is not explained it properly. If I get any proper information about this independent endocytosis, so I will discuss with you. Okay, so thank you, and I will try to exocytosis and the fate of the receptor, what happened just after endocytosis, in my next video. Okay, so continue with my channel.